a good slime mold, right? This is the concept behind why we need to study transcriptional regulation. Slime molds are studied by geneticists, developmental biologists, and others. By the way, those are the red objects, the slime molds, not the moss that's behind it. Slime molds do this really crazy thing. They're initially single-celled organisms. They are individual single-cellular organisms. They're like amoebas. Right? Each cell is its own organism. They can move around. They can take up nutrients. They reproduce and so forth. But they reproduce asexually. They divide and produce daughter cells and so forth. But if they're in a situation where there's a lot of crowding, a lot of amoebas, single-cellular organisms around them, or if food starts to run low, then all of those amoebas start to stream together. They crawl together into a big mound. They become a multicellular organism, essentially. Some of those amoebas, those individual cells, basically give up the ability to have sex and reproduce, and they form a stalk, like a plant stalk, right? A tall structure that builds from the ground and lifts up what you see here, those red parts, those are the fruiting bodies. So in that population of single-celled organisms, when they come together, some of them decide, I'm going to become a stalk, and I'm not going to reproduce anymore. I'm just going to help my friends, the other amoebas. I'm going to lift them up, and those ones that are in the fruiting bodies then can sporulate. They can actually reproduce. And the idea, supposedly, behind that stalk, lifting the fruiting body up, is that a passing insect, mammal, whatever, might rub against it and transfer some spores somewhere else where there might be more food or nutrients where those colonies can start growing again. So this is studied because it's an excellent transition in developmental biology, because this is a single-celled organism that can also decide to become a multicellular organism, like one of these stalks with a fruiting body on top. And these cells, the point for genetics is somehow these cells decide to adopt one fate or the other. When it's time for these fruiting bodies and the stalks to form, how do the cells know which ones are going to be stalks and which ones are going to be fruiting body cells? And most importantly, all of these cells have the same genomes, because it's all the same species. They all used to be amoebas, but they've changed. That's transcriptional and translational control. Because all of these individual cells have the same genes, it's whether or not you turn on the genes that make you stalk and turn off the genes that would make you a fruiting body that makes you a stalk cell. And if you turn off the genes that would make you a stalk cell and turn on the genes that make you a sexually reproducing cell that's in the fruiting body, then you've got a different type of fate. And that's what's called genomic equivalency. That different cells in our bodies have the same genes, but by turning some on and some off, that's how we get to be so complex.